is Emeritus Professor Ulrich Pohl. He's an Emeritus Professor in Cardiovascular Physiology from Ludwig Maximillian University of Munich, Germany. Also a General Secretary of International Union of Physiological Science, IUPS. He's going to talk about regulation of blood flow in the microcirculation. Welcome, Professor.
So, first of all, it will be difficult to cope with such a vibrant presentation. Congratulations. And I also would like to thank the organizers for the invitation, which gives me a chance to my favorite subject in all my scientific life, the microcirculation. And since I got the invitation, I thought I start with an invitation to you. And please forgive me if it sounds very strange, but I want to say, Tafasari, Nifurate, Katika, Majabu, Yamaisha. The translation is, please follow me on my way to the miracles of life. Wow. So I better go there, and nobody can put a note here that I'm over time. And I will start the smallest arteries, and they are divided in the capillaries, and then confluent to the venules, and all these three parts we call the microcirculation. But today we will focus mainly on the arterioles. You see here from our intravital microscopic studies an arteriole that goes into smaller arterioles here, and this is a normal situation. And here you see the situation of an application of an endothelial vasodilator. And you can visually imagine that this is a higher blood flow, better oxygen support, and but this is the exception. This is the exception during heavy exercise. What you normally have is a partly constricted vessel. So we call this the basal tone, and one of the main. And when you electrically stimulate this muscle, then this perfusion unit will react. You will get a higher blood flow due to vasodilation. But this is an isolated effect. Another muscle area will not respond unless you have also activity there. So the whole microcirculation consists of vascular perfusion units. And now we have seen already that with higher workload we get much higher blood flow. And this is shown here as form blood flow, again in units. And now you wonder, what is the main reason for increasing the blood flow? And you may think that it is simply inactivation of sympathetic nervous system. But when you block the sympathetic nervous system, you see that the increase in blood flow is very moderate. During active hyperaemia, you have such an increase in blood flow that sympathetic nervous system cannot explain that. And you see here also an active hyperaemia, and you see just this difference between these two curves, the sympathetic nervous system, and the rest must be something else. So what could it be? It can be local activity-dependent vasodilators. And we often used to call them metabolites. For example, this as potassium plays an important role. This is not metabolite. That's why I prefer to speak of activity-dependent vasodilators. And what do we see here? We have a small arteriole with only one layer of dark, smooth muscle, the endothelial cells within, and the surrounding tissue. Yeah? So what happens now in a muscle when you have exercise? Of course, you have a higher oxygen consumption and you produce more carbon dioxide in the citrate cycle. And you break down ATP, so you have more adenosine. And you have these compounds can come from the tissue, diffuse to the smooth muscle, and relax it. Yeah? If we have not enough blood flow, there will be anaerobic mechanism, and we produce lactate and ions, and we have a decrease in PO2. And this also can directly affect the vascular smooth muscle to relax it. And then we have potassium, and potassium is always released in enhanced amounts in electrically active tissues, like the brain or the muscle, because with every action potential during repolarization, you have an efflux of potassium. And this is so much in highly active tissues that it cannot be removed quickly enough. So for example, in skeletal muscles, the potassium increases from 5 to about 10 in the local tissue. You would die if this would reach your heart, but in muscle, it can do good work, so to speak. Now, these factors here have all specific mechanisms. For example, you see potassium can induce hyperpolarisation, a negative membrane potential in vascular smooth muscle, which prevents influx of calcium and therefore relaxation. And you see nearly all these compounds affect potassium channels. And if you activate the potassium channels, you induce hyperpolarization. There are a few compounds that have also direct effect on L-type calcium channels. And Ray gave us an example for lactate yesterday. And there are also other compounds like ATP that acts on the endothelium and releases nitric oxide, which can work through second messengers like cyclic GMP or cyclic AMA. But all these activity-dependent dilators are very powerful. Most of them work through hyperpolarization in the microcirculation, and that is what we know. And they are the substances that really induce this impressive vasodilatation. Now, however, the question is, since we need to affect all blood vessels all along the perfusion units, how do the signals generated in the distal part of the tissue, how do they reach the more proximal parts? Because all of them have to dilate simultaneously in a coordinated manner. And basically there are two possibilities. One, we could have venous blood draining the tissue and bringing these metabolites via bloodstream to the upstream vessels. That could work in some tissues, but in most tissues, the veins that outflow do not parallel the arteries that inflow. So it is not a good thing to do. The other possibility is that these local tissue activity signals generate local signals in remote vessels, and that this signal is then somehow transferred upstream to induce vasodilatation. And this is indeed the case. You see, again, this drawing, when this muscle tissue is our model, and you see here that we can apply very locally acetylcholine. Acetylcholine is a massive stimulator of the endothelium, and in the microcirculation, it hyperpolarizes endothelial cells. Okay? So if we do that, what do we expect? We expect a local vasodilatation. When we look at this very vessel here, you see this is a rapid transient vasodilatation because we give only a spot of acetylcholine. Now the interesting thing is, if we apply once again acetylcholine here, but look further upstream, you see you have also a vasodilatation. And it happens nearly at the time when, like the local reaction. And if you look further upstream, you see you have also a vasodilatation. So some signal has been transferred via the endothelium, obviously, because this is the hyperpolarization mechanism that is causing that. And we call this the so-called conducted vasodilatation, because it is conducted upstream. Now, I said already that the endothelium acts at the signaling pathway, and the question is, what could be used as the transfer at the signaling pathway? And there we have to look at the vessels, and we have to look at the innermost layer, the endothelium, and this is separated by a basal membrane with holes. This is the gray structure here, and below that is the smooth muscle. And what we see now is that the endothelial cells, schematically, are connected to each other via gap junctions. And gap junctions are places of low electrical resistance. And you see, we have also gap junctions in smooth muscle, and we have even gap junctions, but only in the microcirculation between endothelial cells and the smooth muscle cells. And we have gap junctions are formed of connexins. We have 21 connexins, but only 3 are expressed in the endothelium. Connexin 40, connexin 37, and connexin 42. Now, due to cooperation with Klaus Willicke, we had the chance to use connexin 40 knockout mice. And if something is happening here, and if they play a role, we should see an attenuation of this conductive dilation. This is, I have to go back. But you will see the next peak, or the third next is what I want to show. But I couldn't resist to put this in last evening because it shows you that gap junctions can do an amazing couple of things. These are cultured endothelial cells. They are loaded with a calcium indicator, and blue is low calcium. Yellow and red is high calcium. Now we apply to this endothelial layer histamine. Could you stop? And you see what happens. In nearly no time, all endothelial cells react with an increase in calcium. Yeah? But if you look a little bit more closely, you say, oh no, it doesn't happen at the same time everywhere. There must be something that is transferred. Yeah? And we said, wow, that could be gap junctions. So we blocked the gap junctions with heptanol. Now we go to the next one, please. Okay, and start again. And these are the cells again with histamine. But only a few of them respond. The rest does nothing. So it's the gap junction that harmonizes the whole endothelial layer. And this next slide, please. Thank you. This can be shown if you look in these endothelial cells. All of them have an endothelial knockout, but only half of them have histamine receptors. So it's in fact the gap junctions that control how much these endothelial cells react to histamine. And this is a caveat when you look at marvelous omics, because omics can only average over all cells, and they cannot detect that there are perhaps pacemaker cells, that there's a regulation ongoing, and that perhaps the expression of some gene has little effect because it's not coordinated. Yeah? So be careful if you interpret such studies. Now, I'm coming back now to what I said before. How can acetylcholine induce such a conductive dilation? And what you see here is a muscarinic receptor, and this is work mainly by Steve Siegel, for example, or by some Canadian groups. This is not our work, I refer to it. And you see that acetylcholine is closely coupled with a trip channel, and when we use acetylcholine and binds to the receptor, then this trip channel opens and calcium enters the endothelial cells. And there's an increase in local calcium, and this activated calcium-activatable potassium channels. So when the calcium increases, you have activation of these potassium channels, and the result is hyperpolarisation. Since this vessel, this cell is now hyperpolarized, and this isn't yet, there will be a flow through gap junctions to the neighboring cell. This activates voltage-sensitive inward rectifying potassium channels. You have a hyperpolarization here as well. A hyperpolarization is always the driving force in endothelial cells for calcium to come in. And you have the same game again. You have the activation of the calcium in activatable potassium channels, and so goes it forth and forth. Although there is some attenuation because you lose some of the current through the membrane. There is good work showing that in old people, for example, the setting of these channels is less, and therefore they have less conductive dilation. And this may happen in many other cases. And what happens if you have less conductive dilation? You see the remote responses here in red are reduced, whereas the green responses are always the local responses which we induce at the distant point. And this was the first evidence that one of the endothelial connexins, connexin 40, plays an important role in this conductive dilation. And this work has been done by Witt, who was at that time one of my best co-workers in the lab. He is now independent professor in the north of Germany, but I like to show him in his action as soccer game, and he is a hard player, I can tell you, and he's a hard worker. And now you see also that when you reduce connexin 40 and induce increased blood flow, these are the upper curves here, with increasing stimulation of the muscle, and when you reduce the connexin, or in the connexin knockout mice, you have much reduction of this increased blood flow. So if you have a damage or an inhibition of your gap junctions, you may not be able to work hard again. You are not good in exercising because this is missing. And we are just beginning to understand how this regulation could happen. The muscle where we do our studies is not the only experiment or organ where this happens. These are work from Nelson's lab in America, and they have very sophisticated isolated capillaries from the brain with the adhering or feeding arterioles. Green is the endothelium of the capillaries, and here you see in red the arterioles, the smooth muscle and such. And they apply then with a pipette here potassium Remember, this induces hyperpolarisation. And when they do so, they see upstream here a vasodilatation. This is also a conducted dilation via the endothelium. And you see here, when they stimulate here, they see here a vasodilation, and when they have, they remove one of the potassium channels important for the conductive dilation, they have no dilation anymore. No? So it happens in muscle and brain, but also in many other organs. In the study of Nelson, they think that they're directly stimulating the capillary endothelium. But we must not forget that in the capillaries, you see here a skeletal muscle, there are on top of the endothelium, there are the pericytes. Yeah? And we reasoned that pericytes might be detectors or sensors to affect the calcium influx through L-type calcium channels. Now, it is evident that there must be a lot of regulation. And so far, we know a little bit, mainly from the work of Brent Isaacson at the University of Missouri, but we do not know enough for what's happening here. We have concentrated on another thing. We have looked how many of these myo-endothelial gap junctions are existing in an arteriole. And so we did some work, microscopical work, and you see here, we went through optically through the basal membrane, which is between the endothelium and the smooth muscle, and you see the holes in the basal membrane where potentially myo-endothelial junctions can go through. And we can identify these myo-endothelial junctions because they have an actin core, and this actin core can be made visible. And this, by the way, these lengthy cells, oh sorry, did I switch it off? Or? I see. Okay, I can continue. They can be identified by this actin core, and now we can look whether these gap junctions that are myo-endothelial junctions change their number over time. And there is some spontaneous change, and this spontaneous change that is slow, but it happens. But we found out that, for example, inhibition of an enzyme, AMPK, drastically increases the number of myo-endothelial junctions, and that must have some effect. We have not studied this functionally, but we have found that we can affect the number of myo-endothelial junctions considerably. So there is regular, at that time, with slides, and I had these thick German slides. And they were all used to small slides. And there I gave, and I started my talk, and then the slider blocked. And not only that, all fuses blew. And this was the only time in my life when I gave a full presentation in a totally dark room. So we are a little bit better off here. So the take-home message is the microcirculation is essential for the function of all organs. Without microcirculation, no supply and no life. The second thing is the regulation happens at the level of arterioles. And these arterioles need to respond in a coordinated manner, not just locally. They behave as perfusion units. And to coordinate them under conditions of exercise, we need local activity dilators that hyperpolarize the endothelium. And then we have this conducted signal upstream. And via the myo-endothelial junctions, you have then the relaxation of smooth muscle. And if this does not work, you have a fair reduction of your work capacity. And we are just beginning to understand how we can regulate them. I felt so bad, I could sell so much more. But I had to restrict myself to this conductive dilation today. And I hope you enjoyed it. I have no visually word at the end except asante. Asante sana. Okay. And yeah, I'm ready for discussions later if you want to know more. Okay, thank you very much.